Thanks, everyone. And so I'm in the Department of Physiology and Biophysics, and my collaborator, Dr. Charlie Shoemaker, is in Tufts University. So I don't think we need any introduction. So here, just very simple. Our goal is to use a structure-based rational design platform to develop broadly neutralizing Camelid antibody against SARS-CoV-2. I will just use a few minutes to explain the three keywords here. Number one is why do we need broadly neutralizing antibody? Second is why Camelid antibody and why we need a structure-based approach. So for a lot of coronavirus, and the spike protein is an ideal therapeutic target. So this is a very big protein, and it forms a trimer on the virus surface. This is how they got the name corona, and it is composed of multiple structural domains. So two of its major function is, number one, to recognize the host receptor, and which is mediated by this magenta domain we call receptor binding domain, RBD is here. Then the second major function is to mediate the fusion between the virus membrane and human cell, and that is responsible by this green S2 domain, which is here. So here I show you the crystal structures of ACE2 in complex with the receptor binding domain. Indeed, I superimpose two structures here. One is the RBD from SARS-CoV-2, which is in magenta color, and another one is the original SARS. As you can see, they are almost identical in terms of how they recognize ACE2. So that's why ACE2 is actually a conserved receptor for both of them. And if you look at the interface, there are lots of protein-protein interaction here. ACE2 is here, and this is the RBD of the spike protein. And this motif color in cyan contribute most of the interaction. That's why we also call it the receptor binding motif, RBM. So here I show you actually sequence alignment of 4 representative viruses: CoV-2, SARS, and this is from bat, this is from civet. And we all know the virus somehow jumped from bat and or civet to human. If you look at this area, this is the RBM, receptor binding motif. So amino acids in this area actually change a lot. And based on the literature, we all believe the spike protein is under positive selection pressure, especially in this area. And actually, mutations have already been identified in human, which showed up in this area. So this actually readily explains the notion that there are many neutralizing antibodies against SARS, but they don't work for SARS-2, CoV-2. So I show you 2 examples. One is this called ADR, and it binds here. This is the RBM. Another one, M396, binds here. And if you look at the interface, the epitope, so ADR is this circle. So that's the epitope on the spike proteins here and here. And for M396 is this plus sign here, here, and then a last here. So you can see the epitope for those 2 antibodies are very different between the SARS and SARS-CoV-2. That's why they don't cross. But still, we have hope. If you look at another one called CR3022, the structures was recently done in the Ian Wilson lab and published in Science. It binds here, so it has nothing to do with RBM, so it doesn't compete with receptor binding. And that's why it doesn't neutralize. But if you look at the epitope, represented by the stars, so the epitopes of it is outside the RBM. And you can see these regions is actually quite conserved. So that's why you end up has a dilemma here. On the one hand, you have good antibodies, potently neutralize SARS, but they don't cross. Then you have antibodies. It could cross, but doesn't neutralize. So an ideal antibody we want is it binds here to compete and neutralize the virus, and also could bind conserved epitope, so to be able to cross. But unfortunately, this is a huge area on the spike protein surface. No single antibody is able to do so. That actually comes to the second point, why Camelid antibody? So this is a New York Times report just last week. A llama named Winter got famous because it produced an antibody that could neutralize both SARS and SARS-CoV-2. And the paper was published very recently in Cell. So we all know very much about IgG, and we like it. Very good, very powerful, but we know it's very expensive to produce. And Camelid actually produced a special type of antibody. It only has a heavy chain. That's why it's called a heavy chain only antibody. And this is the VH domain of the heavy chain only antibody. That's why it's called VH of H. And someone also call it nanobody or single domain antibody. And in the recent years, VHH has gotten more and more popular. For example, a company working on VHH was bought by Sanofi for almost $5 billion in 2018. Then a year later, their first drug was approved by FDA. Like everything else, the Camelid antibody have their pros and cons. So they are very small and easy to modify, and they could target some unusual epitope that are sometimes inaccessible to the regular IgG. They have low immunogenicity, and they are easy to produce and formulate compared to IgG, and they can be nebulized and administered via an inhaler, which is make it very unique for the treatment of respiratory disease like SARS. They are very stable, can be stockpiled, and they can be encoded in RNA for delivery. And some of the disadvantage includes like possible immunogenicity, and which could be overcome by humanization. And because they are small and they may have low affinity, and they have shorter serine half-life, which could be overcome by fusion with an Fc tag. So why we need structure-based design? So this is the traditional way. And a good example is in this recent Cell paper, and which was actually drew a lot of media attention. So they have to take spike protein from different viruses like SARS or SARS-CoV-2, the immunized animal. Then they take all the VHH they could get, and then they screen. And they look for any VHH that could cross-protect, say, SARS and SARS-CoV-2. And they found one. And in this case, structural biology is not necessary in their case because they knew the result already before they work on the structure, but they need the structure information to guide their optimization and also understand the mechanism. But we think we need something which is more rational based and we have more control. That's why we propose this structure-based platform. So the step one is the same, and we will immunize animals and get as many VHH we could get. But then we will take those VHH which have distinct epitopes, meaning they do not compete with each other. Then we mix all the VHH, as many as we could, mix them with a trimer spike protein. You can imagine we can get a super complex with a trimer spike protein with VHH decorated everywhere on the surface. Then we bring this super complex to a national cryo-EM facility. This one we're working with is in Oregon, and we have an active proposal with them. So we got to use their $1 million microscope for free. Then we will get all the structures at once, which will tell us a large number of surface epitope on the cell, on the spike protein surface. Then we will separate our VHH into 4 groups based on 2 criteria. Number 1, do they block ACE2 binding? Meaning, do they neutralize or not? Second, we will combine the structure and the primary sequence alignment to ask whether their epitopes are conserved among either SARS-CoV-2 or the CIVID bat viruses. So you will get either neutralizing and conserved, or neutralizing, not conserved, or non-neutralizing, conserved, or nothing here. Based on that, we are going to design a molecule we call VHH-based neutralizing agent, VNA. VNA has 2 arms, and the neutralizing arm is 1 VHH molecule, which is very small, about 12 kilodalton, and it could be a VHH in this group or this group. And the anchoring arm is another VHH, could be one from here or here. Then, because we know the structure of them, we know where they are on the surface, we know the distance, their orientation, and we are able to design an appropriate peptide linker to connect them. So both of them can simultaneously either bind to a one spike protein monomer, or they could bind 2 spike protein, neighboring spike protein in a trimer. So they can bind simultaneously. And we believe the synergy between those 2 will significantly increase affinity and also broaden the protection spectrum. Let's say in the future, a mutation may happen in a patient in the epitope of this nanobody. So the affinity for this guy decreased, but then the anchoring arm, which hold on a conserved epitope, will still be able to provide strong support. So the VNA as a whole could still maintain a high affinity and still be able to neutralize. So, and this platform actually has been validated in our other project against botulinum neurotoxin. And this is a CDC tier 1 select agent and a potential bioweapon. So you can see, we actually just published a paper this year, and you can see this example. We have multiple VHH bind to the toxin surface. And then in this experiment, we designed a VNA, which is composed of 2 VHH with a linker, and they can bind simultaneously. And our VNA can very potently protect the A-type and B-type toxin up to 10,000 fold LD50. And in this control, we use the same 2 VHH, but we link them in a way they cannot bind simultaneously, and it doesn't do much. So it's just a good timing that, and we published that and just like 2 months ago, and now we're going to use that for a new target. Okay, that actually ends my talk today, and we'll be happy to answer questions.
This is Don Forthall. I had a question.
Sure.
A lot of virus neutralizing antibodies target fusion rather than virus attachment. And so I'm wondering if you might not consider, in addition to targeting the receptor binding domain, that you might want to consider targeting S2 as well, which also would be more conserved.
Yeah, that's a very good question. You actually, I almost wonder whether you just read my R21 proposal. Yeah. So and when we wrote this one, and this is a very, we are very grateful to get this funding. But again, we are proposing the huge amount of workload here. So to be honest, and you know, we don't really know how much we can achieve here. That's why we picked this ACE2 as one point. But then we are applying other funding. So in the new proposal, we are actually targeting both. So we are going to, and so the project will be the same. We will solve all the structures at the same time. But the difference is we will add the second R. That is to look for VHH that actually interfere with the S2 function, which is the conformational, either block the S2 conformational change or block its membrane insertion or the membrane fusion. So that will be a separate type of neutralized antibody.
Good.
And also the S2 region is more conserved than S1. But you know, for some reason, most of the known antibody for original SARS are all blocking the ACE2 binding. So there may be a reason, maybe ACE2 blocking would be more efficient. But as you said, we believe S2 is equally important in terms of neutralization.
I think, Rongsheng, it's good to look back at some of the older data, because a number of us have targeted the fusion domain, have targeted S1 and S2 as long ago as 1982. And there are some interesting outcomes of that. If you target, for example, the epitopes that are in S1, the virus is capable, at least in the mouse hepatitis version, which is a close relative of this virus, is able to delete up to 152 amino acids worth of sequence to get rid of that epitope. And that affects both CD4 and CD8 epitopes. So, you know, there's a lot that can be learned about predictive behavior in these. One of the best neutralizing antibodies that we had was also a fusion inhibiting antibody. So you're on the right track there. So there's a lot of things. If you're interested, I can point to some of those older papers.
Well, that's definitely. Yeah, I think that's also the good things about the university is putting everything together. I think here, as obviously, we heavily focus on the in vitro study. So we really want to have collaboration with either the virologist or immunologist. So, and to know more about that, and also, you know, to put potential the new VHH into either as therapy or even, I didn't mention here, that could be also very good targets for detection. And because those VHH has high affinity and high specificity. So, you know, and that would be, there are companies already, they want to use that for, to generating kits for screening. And so that would be fantastic to collaborate.
Yeah, I think the other thing is, as far as the Camelid antibodies, look at this at the literature that was generated on MERS, because there was quite a bit of work that was done on Camelid antibodies in MERS.
Okay, good, good.